And then there are many transmembrane protein tools. So this one probably is the most popular. That is called HMMM. It's called transmembrane hidden Markov model. You will see from name, actually, initially it was a hidden Markov model for transmembrane prediction. But then pretty much all the hidden Markov model for bioinformatics are replaced by deep learning methods. So they also have a deep learning version, which I'm going to do a brief demo. And then here you see that this tool can predict the transmembrane segments. And then you can predict the so-called orientation. So this orientation cannot be predicted by AlphaFold. That is which side is the inner membrane, which is the outer. Let's say on the surface protein, which side is extracellular, which side facing the cytosol. This actually is very important. So such kind of tools can predict such, this orientation or topology. And then these two can also compare same transmembrane protein in different species. You can see which part is conserved, which part is not conserved. So this is a very useful tool. It has been cited for more than 13,000 times. So we'll have a demo of this tool. And then there are a database about the orientation of protein and also classification of a transmembrane protein. It's called Orientations of Proteins in Membranes, the OPM database. So as I mentioned, it's this helices or overall protein. They do not stay perpendicular to membrane. In many cases, they have an angle. So this angle is driven by this polar structure of this helix or membrane. That is, you have the charge, you have this dipole in electrical charge. So that's why it has to tilt a little bit in order to offset the charge balance. And then this can be predicted using some computational tools, mostly based on this transfer energy. That is, if you transfer a protein from water into membrane, what is the energy difference? Of course, you want to minimize the energy in the transmembrane. So that's how this orientation can be calculated, and it's actually pretty accurate. And then transmembrane, this database also classify it in very details. So this is also very useful. Another type of a transmembrane protein is a beta barrel. So if you look at this, so this is a beta barrel for the transmembrane region, and then the outside of the membrane, there could be a lot of helices. But within the membrane, it can be either barrel or helices. It cannot be mixed. But the same protein inside and outside, it can be mixed. So if you see from top, actually it's a beta barrel, there's a big hole in it. So that's why it can be used as a transporter. So this is this is one example of this beta barrel called pyrines. It's a well-known protein. And then beta barrel transmembrane proteins also can have a different number of beta strands. So here are some common ones. So it's 8 strand, and then 12 strand also common, and could be a lot bigger, like 16, 22. So could be really big. This beta barrel proteins. So beta barrel is similar like alpha helix transmembrane protein. So it has tools to predict just a beta barrel. Unfortunately, beta barrel cannot be predicted as accurate as alpha helical transmembrane protein. Alpha helical transmembrane protein having more than 90% accuracy. They're very accurate. Beta per barrel, at least 10% less accurate. But these two are still very useful because even though you can use AlphaFold to get the structure, these two can give you complementary information. And AlphaFold may not be 100% accurate either. So this information is complementary. More importantly, these two can predict the orientation, which is inside the cell, which is outside the cell. This tool is also predicted using hidden marker model. I haven't seen a deep learning version of that. I assume someone eventually will develop a deep learning version for this type of prediction. But this tool also widely used, even though it's not as widely used as the alpha helical transmembrane protein, but still it is useful. So I'm going to do a few demo. You can see this how they perform. Okay, so let's see this this is called the OPM, orientations of proteins in membrane. I think the first useful information of this resource is this really classification. So you see, you actually classify proteins in many groups. So this you can see this is a transmembrane. You see different classes of transmembrane proteins, and then there are 11 classes like alpha helical, and then there's some others like a beta. You see beta hairpin, beta helical, etc. And then you can go down to each one of them, and then all the way to individual protein. So for the individual protein, you see what kind of arrangement it has. And then also this the tilt, in this case, is actually perpendicular to transmembrane, to the membrane. So basically park on the surface of the membrane. And then some other cases, you can see alpha helical. So let's just see some alpha helical proteins. For example, this one. Yeah. So here, it tells you this angle. So this angle basically means the overall protein, the main axis of the protein. In this case, it's tilted by 12 degrees. And this is the transfer energy we talked about. That is from water to membrane. Energy change is at 96 kilocalories per mole. It's actually huge compared to a typical globular protein folding. This actually is a lot bigger. And then you also see some other information. So this is this OPM resource. So if you want to know your transmembrane protein in terms of context, then you can look at this. Of course, many of the protein sequences, you do not know the structure. But nowadays, you can use AlphaFold to predict structure, and then use structure comparison to find one of the known structures. And then you come here, you see, let's say, this known structure. Based on information here, you can infer your protein. So that's kind of one way you can use this resource, even you do not have the structure. So here show this web resource we talk about, is deep TM HMM. This is actually very simple to use. So I just copy the protein sequence here. So this is a protein. Yeah, I talked about it earlier. This is a protein in rice. So you see, it's very easy to use. That means you just do copy and paste, and you run it. Yeah, typically this kind of tool, so it's very fast. You can just wait. Just in real time, you get the result. So So you can also run this tool through command line. You can download it and then use it for so-called batch mode. That means you run it for many proteins simultaneously. So here is still running. But I said this tool has been widely used for so many years. I think that's really a role model for bioinformatics tools. That is, you start something, you keep maintaining it, keep revising it. Eventually, it will be pretty good. Okay, maybe we'll just let it run for a while. When I tried last time, it took like less than 10 seconds. So it could really vary. This tool is for the beta viral protein prediction. It's called a PRED-TMBB. So you can go to the site, and then you just paste the sequence. So it just found the protein. And then you can have different methods. But let's say just use the default method, and then you can run it. So this one actually runs pretty fast. So you see here, this is the beta barrel, and then it tells you where are the beta barrels, and these are the regional beta barrels. But also it tells you which one is the outer, which one is the inner. So the blue one is the outer. That means if it's let's say on the cell surface, so blue one basically means the extracellular component of that loop. So basically, transmembranes are segments that connected by loops. So that basically means that loop is outside the cell. And then if you see green, that means it's inside the cell. So this information sometimes actually pretty critical. For example, you want to design a drug, you want to target the extracellular components, not the intercellular component. Okay, and then here, of course, you can generate like a publication quality images, and then you can just use it directly in your paper, provided they have the copyright information. Okay, so this one is still running. Yeah, so I don't know. It should be pretty fast. Maybe I better just resubmit. Okay. Sometimes you resubmit, it could be even faster. Yeah. Okay, so maybe today is a little bit slow, but basically you will see similar information as I showed for beta barrel prediction.